Thank you very much. Good morning, guten Morgen. Very happy that many people showed up to join our webinar entitled How to Improve Your CRISPR Genome Editing Experiments. So we missed a slide. I should have moved forward a little bit. I'm going to very quickly, for those who do not know IDT, very quickly introduce you to the company and how we got to work in this CRISPR area. IDT is the largest oligonucleotide manufacturer worldwide. We have over 82,000 active customers, which are customers who ordered in the last 3 to 6 months. On average, we manufacture about 55 unique sequences, as well DNA, RNA, modified, purified sequences, so the entire portfolio, which is about slightly over 50 megabases of DNA that we synthesize. Here you see a picture of one of our production halls in our headquarters in Iowa. And as you can see, there's only very few people working, meaning that our synthesis platforms has a very high level of automation, which is actually discarding the possibility of introducing human error. And that is a lot of quality control that we actually enter, and which is necessary to guarantee the processes and making sure that the quality of the sequences that are made are actually at the level of the research that end users are performing. IDT is a global company. So we have our headquarters in Coralville, Iowa. We started as a spin-out company from the University of Iowa. We have offices, and it's also where our CEO is located, in Skokie, Illinois, which is a suburb from Chicago. We started producing in 2006 in San Diego. In 2008, for the European market, we started a production facility in Leuven, Belgium, where I'm based. 2012, we opened a production facility in Singapore, and since last year, we have an R&D center in the Bay Area, where a lot of our efforts in mainly next-generation sequencing and qPCR are actually performed. Now, what is the secret of IDT? How did we become a world leader in oligosynthesis, which is actually used for CRISPR applications as well? It's actually based on the quality of the product that we are capable of leveraging. What we did was to try to improve the synthesis and the quality of chemical synthesis of DNA oligos. Here in red you see what the average industry standard is in terms of the coupling efficiency. Now, what is the coupling efficiency? It's actually the percent, when you make DNA or RNA sequences, you're actually always adding one base to an existing sequences until the moment that you have the required sequence. And so the average coupling efficiency in the market is around 98.5%. And what we did, and you saw that on the picture, is we developed a synthesis platform which has mainly as goal has to increase that coupling efficiency. So currently for our standard oligonucleotides, we achieve routinely 99.25% coupling efficiency, and for our ultramer, those are the very long sequences that we need to make, we are capable of reaching 99.5%. Now, how should you interpret these coupling efficiencies? When you make sequences of about 10 or 20 bases, you see here the full-length product over the oligonucleotide length. for a 20 base working with industry standard coupling efficiency, you would have on average around 75% correct sequences. The other 25 are truncated products. With our synthesis platform, we're actually still higher than 85%. Now, this is only for smaller sequences. As some of you might already know, sequences mainly used for CRISPR applications are very long sequences, and so we're there in the range of actually, as you will see later in the presentation, it starts at around 89, 90, even up to 120, 140, and so you see that the discrepancy that you get there just based on oligosynthesis plays a huge role in the quality of the final product that you are getting. Now, next to the synthesis, the improved synthesis, a second advantage is that we have a very precise way of performing the quality control. We use electron spray ionization, which is a technology 15 times more sensitive than Malditov, which is considered the industry standard for oligosynthesis. So the quality of the product that we have available, the way that we make DNA and RNA synthesis is what made IDT from a small spin-out company to the level of the global player that we are right now. Now, more recently, what we did was to leverage that our advantage in synthesis of DNA and RNA fragments and actually develop solutions to the market. So as you can see, we are active in fields like qPCR, synthetic biology, we make RNAi sequences, we have the regular qPCR probes, but also probes for genotyping available. And so more recently, we also went into the CRISPR field, and that will be the main focus of this webinar here today. Now, I'm not going to tell you the entire background of the working of CRISPR-Cas9 system in bacteria, and how it was developed in a product like how people are using it right now. What I will do is I will just give a simple overview of what CRISPR-Cas9 is used for those who are brand new to it and want to discover what it is. So basically, CRISPR-Cas9 is a programmable restriction enzyme or programmable nuclease, which is composed of a protein nuclease called Cas9, which comes from bacterial origin, and which is driven by RNA molecules. Now, this can be in a natural system 2 RNA molecules, and those have been engineered so that they can also be used as one single guide RNA. We'll come back to that in one of the next slides. The complex, the Cas9 CRISPR complex, they will actually scan along the DNA. It will stop at a specific regular motif, which is called the NGG motif. It's also called the PAM site, which stands for protospacer adjacent motif. Now, this allows the enzyme to unravel the DNA. And if there is a match between the RNA that drives the reaction and the target DNA from the cell, the enzyme will cut the DNA in a double-stranded fashion, causing a break. Now, a break in DNA is actually toxic for the cell, so the cell has to repair it, or you could say it's going to die. Now, the most common way for the cell to repair is by non-homologous end pairing, which is basically just putting the 2 pieces next to each other and fuse them together. If that is done and the repair is perfect, the original target sequences is reconstituted. Then the enzyme comes in and it cuts it down all over again. The only way to have a permanent effect is to modify it slightly so that the enzyme cannot cut again. That is basically used for gene disruption. And so people who target a gene they're interested in, they will create some modification resulting in a prime shift mutation and rendering the expression of the protein non-functional. Another interesting pathway uses homologous recombination, and where you provide actually some donor DNA which has overlapping sequences to the site you want to cut, and through this mechanism you can actually integrate this donor DNA into the genomic DNA of your cell. This is a seamless integration, so you can have from a few bases, which you could actually use a long oligo for, like the ultramer oligos that we have available, or it can be a complete pathway where you need, it's a little bit trickier because therefore you need slightly longer overlaps. Here is representation of the two main systems to drive the CRISPR-Cas9 reaction. The common part in both is the enzyme. And so one of the great advances that the Downey lab, where you see the reference of the paper over here, was that they understood, and actually they confirmed as well, that you could fuse those two RNA oligos together using a linking loop. That reaction now, using one sequence, also called single guide RNA, it has a 20 nucleotide, 20 base pair complementary sequence to the target sequence here, which is specific then for your experiment. And then we have this extended tail of RNA, which is called the tracer RNA, which in the natural system it's called, tracer RNA. And that one, this entire RNA system will drive the reaction and activate it. The two-part CRISPR RNA tracer RNA system is the way that it's actually available in nature. It's the natural bacterial system. And it consists of two RNA sequences. So the one, as you can see here in green, is the CRISPR RNA, and the separate is the tracer RNA, which is longer than the CRISPR RNA. We'll give the details a little bit later. So in between those two systems, besides this loop here, there is not much difference, and the overall mechanism actually used in both cases is really similar. Here is a representation of the first CRISPR experiments that we performed at IDT. So what we did was we purchased two plasmids from different vendors, and we transfected them in the cells after engineering the target site. And really what we did was to see just what happens. So what we have here are plasmids with, in this case here, the single guide RNA and the Cas9 expressed together. Now what you need to realize in such a dual expression plasmid, which is commonly used in the CRISPR field, because one of the advantages, it's very cheaply offered through the company Adgene. But what you need to know is that those plasmids are very large. Actually, they're often in the range of 7 to, in this case here, it's 9.2 kb, so they're very often between 7 and 9.5 kb. Now, for those having experience with it, you all confirm that common to large plasmids is the fact that those are really difficult to transfect. As we see here in the picture here under, we approximately only had about 10% of our cells that got transfected. when we compare that to a smaller plasmid, in this case it's an almost 5 kb plasmid expressing the EGFP gene, we saw transfection efficiencies which went up to 90% in the same cells with the same transfection specification. So this very minor experiment actually shows one of the starting major difficulties of CRISPR, and when you need to work with the plasmids for driving the CRISPR reaction, and that is that you will have quite low to actually extremely low transfection efficiency. As from the start, we didn't feel comfortable working with such large plasmids, and to actually add to the complexity of it, it's not only that you have a low transfection efficiency, but the transfection efficiency on itself is highly variable from day to day. So some days we obtained transfection efficiency of 10%, and the next day it was about 25%. So it was really complicated for us to compare experiment to experiment. Our aim was to develop triggers and look at the chemistry, but we quickly realized that plasmids wouldn't allow that due to transfection efficiency and the variation, as mentioned before. So in first instance, we decided to develop our own cell line. So we have an HEC293 cell line which expresses the Cas9. And as a side note, of the two plasmids we bought, one worked well, the other one not that well. Actually, when we sequenced everything, we found that there were 13 mutations. And that is not a problem of working with plasmids. It's not always what you expect it to be. On the right side here, what you see is a Western blot, and we looked at the presence of Cas9 in the transfected cells. So what we used here was whole cell extract, not purified. So we noticed that out of the only 10% of the cells that were transfected, the amount of Cas9 that was expressed was absolutely huge. Here we compare it to beta-actin and HBRT1. So one of the issues there is that you have an overabundance of Cas9, whereas the transfection efficiency is really low. As mentioned, we developed our own cell line. So we selected a few cell lines going from low expression, mid and high expression, and we picked a low expressing cell line because very basically for us it was the one that worked best. If you look at the gel here, you can see our Cas9 express cell line expressing much lower amount of Cas9 than the cells that were transfected with the plasmid. And that is an important and a typical feature, as we believe that the presence of too much Cas9 will not only enhance but actually will cause off-target effects. The main reason why people are worried about overexpression of Cas9 is that you have a lot of protein in the cell, and typically there's only a low amount of targets available in the cell. So you are stuck in a situation where you have a lot of protein that, to say it in a very simple way, has nothing to do, and so will most probably pick other DNA and RNA sequences, causing these unwanted off-target effects. Here is an overview of the different techniques to drive the CRISPR-Cas9 reactions. At first, it looks very complex, as there are many ways to actually perform CRISPR-Cas9 experiments, which also indicates, I think, that it's a quite young technology and people have been searching for let's say, the right way to do it. So you have dual expression plasmids. You can have the Cas9 and the single guide RNA expression plasmids, both in separate plasmids as well. You could use Cas9 mRNA. We and others also derived Cas9 stable cell line for screening activities, but more recently people actually, to obtain the protein, people have started to use the protein for the RNA trigger. People have been using plasmids in the past as well, right? Right now we also sell, to get the sequence in, a lot of G-blocks gene fragments as single guide RNA. These G-block gene fragments are linear double-stranded DNA fragments that can be obtained up to 2 kb. One of the advantages is that we can synthesize those in a very fast and in a very cheap way. They are designed by the researcher to include a promoter. In this case, it's a U6 promoter, and then the single guide RNA. You can clone those G-blocks in your expression plasmid, but you can also transfect them directly and drive the reaction. So its transfection is actually very easy. A lot of people have also started with using single guide RNA expressed as IVTs. The reason for that is that the problem of working with plasmids and G-blocks is transfection, because researchers need to wait for them to be expressed and then delivered to the nucleus before they get active. And the advantage of working with IVT-generated single guide RNA is that they are active in a much faster fashion. And then finally, there is the chemically synthesized RNA sequences, which is actually the system how IDT developed it. What we did is we went back to nature and synthesized and engineered those RNAs to be more efficient, which will be actually the main topic of this presentation. A few words about single guide RNA made by IVT. IDT has a lot of experience in introducing RNA and DNA sequences in the cell. So we performed a lot of work in RNAi field, in work with antisense oligos. And we know from that long history that introducing long RNA oligos is a bit tricky. Why? Because long RNA oligos are toxic to the cells. As you can see here in the middle picture, which is single guide RNA injected, and which was made by IVTs. These were made ourselves, but we got the same results with single guide RNA expressed as IVTs purchased by third parties. Compared with the other two, we have non-infected ACT293 cells and our Twopart system. We see that there was very fewer effects compared to the long single guide RNA. We'll talk about our Twopart system just in a minute. Now, where is that toxicity actually coming from? Actually, it's related to the innate immune response, which actually triggers a cascade of genes. So some people will say that their cells are not immunogenic, but what they forget is that their cells still have the genes. Those genes are within the DNA. And in certain circumstances, it can induce parts of the complete pathway. So depending on the type of cells that you're working with, so we have performed most of our work with HEC293 cells or HeLa cells. Those are quite stable, so actually meaning that we lost part of the pathway. They're not going to die, but they just might look a little sick, to say it that way. When we go to more complex cells, primary cells, for example, those cells are much more affected. This is feedback that we get from the field and from our collaborators worldwide. And they see, of course, major benefits in actually avoiding working with IVT-generated single-guide RNA because that immune response. And so they seem to have like a preference to switch to a system using shorter RNA sequences. So what we do here is we look at a couple of the markers, these three, for example, which here had shown a similarly high induction when treated with the single guide RNA expressed as IVT, whereas no inductions were detected when we treated it with the two-part RNA oligos, which is small, which I will show you on one of the next slides. So we understand that some people prefer to work with the cell lines that they have and that they know best, but you need to consider if you have a high mortality percentage, which could go up to like 60%, you really jeopardize your experiments, and at that point we could actually advise you to consider for alternatives. We've been talking about the single guide RNAs as which can be introduced as expressed by IVTs or as G-blocks. Now we're going to look into the natural two-part systems as you can see it in bacteria, which, as mentioned before, consists of a CRISPR RNA sequence and a tracer RNA sequence. When we look at the native sequence, the natural CRISPR RNA is about 42 bases long, right? Which is actually not too bad to introduce into cells. 42 bases is not considered immunogenic, but just from a technical point of view, for us as a company, the shorter that we can make them, the better it is, because there is it is easier from a manufacturing perspective. You can see here that the tracer RNA is actually even more problematic, because in the natural occurring system, that tracer RNA is about 89 bases. 89 bases is really long for RNA oligos. This could be immunogenic. What is the reason? As you can see here, what we try to do is to make them shorter, is just because synthesis of such a long fragment is really complicated. And so we wanted to make them shorter because that would actually influence as well the turnaround time as the cost of making the tracer RNA. It is universal, so for all the CRISPR experiments it's the same sequence, 89 bases long. It is close to the limit of RNA oligos. So due to the fact that it is universal, what we did is what we considered to make huge amounts of this sequence. The advantage for a bulk synthesis like that is that we also avoid batch-to-batch variation, which has an influence on the cost, and that's always important when performing experiments. But for the tracer RNA, as you can see for this length, there is for us a real big incentive to make them shorter. And so we adapted the sequences, we tried to make them shorter, and shortest to the the point where we actually lost activity. On this slide here, we look at how this is actually a walk through the experiments that we performed, and it was really what we tried to do is to look at several variations from the end. So we looked at the structure and looked at what the possible modifications could be that we could do to the sequence. On the right here, you see the corresponding activity with the adapted sequences. So on top here, the first one is the naturally occurring system. And you can see that some of these variations actually increase the potency of the molecule. But also what you can see is sometimes that like changes of only a couple of bases, 1 or 2 bases, completely kills the activity, which therefore has dramatic effects. And so we needed to find the compromise between going short and keeping the activity of the tracer RNA. Next to the sequences on the outside, we also looked at potential internal deletions, and from what we knew about the molecules, also what we read in literature, and just by talking to our collaborators, we came to the conclusion that actually changing some of the internal structure of the tracer RNA highly compromises the efficiency of the sequences. So here you can see that on this slide, but what we looked at is here we really designed the tracer RNA sequences and then just tried to modify internally to figure out which were those parts of the sequence that were really crucial for the work performed with CRISPR RNA sequence. So we did actually a combination of several hundreds of candidates, and here is an illustration of what we did in terms of length optimization of as well the CRISPR RNA and the tracer RNA. So bottom line, our goal was to make them as short as possible without jeopardizing the activity. We gradually decreased the length of the tracer RNA, right, as you can see here. So we went from the natural 89 nucleotide tracer sequence down to 74, 70, 67, 65, 63. And also combining that with decreasing CRISPR RNA sequences, changing from 42, the naturally occurring system, down to 34. And we could come to a compromise that we use for the further development of our two-part system, where we use a 67 nucleotide tracer RNA combined with the 36 nucleotide CRISPR RNA, which a little bit, I wouldn't say surprisingly enough, actually gave better performance, not just equal performance, but better performance. So we have the advantage of easier synthesis, but also of higher performance, higher cleavage, better efficiency of genome editing. Now, just a few words about the assay that we use to measure the efficiency of genome editing. Most of the results, unless we sequenced the fragments, we used the T7 endonuclease mismatch assay, which is actually an enzyme that we currently ourselves don't sell, but which is available through the company New England Biolabs, for example. It's far from being perfect, but it gives you a very quick indication, and it allows you to compare the results from one experiment to the other. So what the process is you design actually PCR primers. You perform PCR amplification around your cutting site, which after genome editing will actually have a number of different modifications associated to it. So you will have the wild type, but you also will have insertions and deletions. Once the PCR is done, you heat it to mix them up, and you will partially form heteroduplexes, which you will submit to the endonuclease, and this endonuclease is going to cut everything with a mismatch of more than one base pair in length. So the enzyme doesn't work with one base pair, so that means that you will always get an underestimation. So as you can see over here, it does not detect or it does not cut actually one base pair mutations, but you are capable of actually looking at the cuts which are generated by mismatches of more than one BP. So some devices can actually calculate back the percentage of genome editing. One that we're really fond of working with is the device, the fragment analyzer from the company Advanced Analytical. It's very easy. It has a 96 well setup, and you can load up to 10 plates at once. So there is 1 hour. It takes about 1 hour to read one plate, but it's fully automatic, so it's actually quite convenient to work with. The result is actually a pseudo-gel readout, and the machine does actually all the calculations for you to get an immediate readout. So very convenient, allows to go through hundreds of genome editing events per week. So a couple of words about the assay. Here is actually a broader view of a comparison between the natural CRISPR-Cas9 system. So remember, that is one with the longer sequences, and our two-part system using shorter CRISPR RNA and CRISPR tracer RNA sequences. We're looking here at 12 sites in the HPRT gene, but actually we did this experiment for many genes and many sites. And as you can see, the shorter version actually consistently triggers a significantly better genome editing than the naturally occurring system. Even in the natural occurring system, some sites were pretty well, but it is obvious and systematic that the engineered system works much better than the naturally occurring. So a couple of words about that two-part system that we developed. So the length optimization was basically started because of the easiness of manufacturing, but we were capable of actually getting a better result with that. So when we do a comparison, the natural length is about 42 bases. We got that down to 36 bases. For the tracer RNA, we were capable of drastically decreasing the length from 89 bases to 67 bases. This system is therefore actually more affordable, easier to make, gets very good transfection efficiencies. And we also looked at chemical modifications for increased nuclease resistance. We come from that because of our long history in RNAi. RNAi as well, we entered chemical modifications that will add some desired feature, and that is not possible when you work with single guide RNA or as well as with G-blocks as single guide RNA that is expressed by IVTs. So before going into detail of the chemical optimization, I want to talk a little bit about how efficient CRISPR is. What we did here is we tested every single PAM site, so every single NGG sequence in a number of exons of various genes. So, for instance, we had a couple with high GC content, this one here, 62% GC content, and we see that actually 95% of the sites work really well. Work well, between brackets, can be defined as a genome editing efficiency of about, let's say, 20% cleaving according to the fragment analyzer. So this slide here is showing that there are only very few sites for this gene that work, let's say, poorly. When we compare this CRISPR to a technique like RNAi, there you only have like approximately 1 chance in 4 to have something that works really well. So in that case, genome editing is much more efficient, where you have almost 3 out of the 4 sequences that you are going to design will work well. Now, the importance is to find the right site, and of course also to focus on potential off-target effects. To measure off-target effects, or at least to predict them, we use the tool from the MIT. Actually, you can very easily Google it. If you Google MIT and CRISPR design, you get to the design software developed by the Zen group. It's fairly easy to use. You just need to enter a couple of bases of the target region that you want to design for, and it gives you actually a pretty detailed report on the potential of target sites. Now, to come back to this slide here, we also selected some genes which were GC poor. This particular case here, we have only 38% GC content, and actually the lowest genome editing efficiency that we had was for the STAT3 gene, and that was only 75%. Bear in mind, and very important to consider, is what I mentioned, the T7 endonuclease assay, which was used to actually measure the CRISPR efficiency, is always an underestimation. So reducing further the number of sites that actually don't work, so the efficiency that you actually obtain is higher than what you see over here. So bottom line, what you would like to do when you work with a couple of sites is you want to make sure that you work with sites that have very high efficiency and that are predicted to give the least possible off-target effects when designing them. As we say, one of the ways to do so is using the Zen software. Now, we know that RNAs in cells can be protected to some degree, but when performing it in vivo experiment, you risk to have degradation by nucleases, and that can strongly influence the reaction. So, can chemical modifications improve the function of your CRISPR RNA oligos? Yes, of course. Our long history in modifying RNA oligos have actually led us to the development of these chemically modified sequences. And the nice part as an oligonucleotide manufacturer is that there is actually a pretty large but basic toolbox available for modifying RNA sequence. So the most commonly used are 2'-methyl modifications, phosphotyrate bonds. Slightly more exotic, I would say, is a 2'-fluoro modification. LNA could be used, or eventually end blocking groups. This is a publication by the company formerly known as ISIS. Their primarily work was to look at modifications to see if they could make the RNAs more stable, and so we also went down that way. So when we study chemical modifications for the CRISPR system, we have, as mentioned before, we work with our two-part system, which consists of a 67 tracer RNA and a 36 CRISPR RNA. We have for the tracer RNA, we have a sequence that has 67 bases. That means 66 linkages and 2 ends. Or this allows us to have 135 places to modify. For the CRISPR RNA, we have 36 bases. That means 35 linkages and 2 ends to modify, which in total gives 73 places. When you multiply those 235 and 73, that means we have thousands of possibilities, but of course we narrow that down slightly based on the experience that we have. This slide here is just an illustration of the work that we did. In terms of proprietary technology, of course, it does not contain the final variation that we have available in our ALTAR system, but I just want to give you an idea of what we did to actually come to a consensus of the best working and most stable sequences used for CRISPR. The first one here that you see is the natural occurring tracer RNA system, and what we did then was just trying to implement several modifications. So first of all, when we compared on the activity, what we did is, if we try to completely adapt the sequence using 2'-O-methyl. Now, 2'-O-methyl typically blocks endonucleases, but as you can see, switching all the bases to 2'-O-methyl completely killed the activity of the CRISPR-Cas9. We also looked at introducing phosphotyrate bonds at the end, and those modifications actually, they block the exonucleases, which are, as you might know, an issue in most of the systems. And so you see that from the modification pattern that we use, that sometimes you see that you have a similar activity, which is what we accept. So bear in mind, what we try to do here is not to increase the efficiency, but actually to come to sequences that are much more stable and less susceptible for being cut by exo and endonucleases. So in some cases, we just saw that adding one more modification, so in this particular case, or in this particular case, just one more modification drastically decreased the genome editing efficiency to almost kill it completely. We did the same thing. We did the same thing in the CRISPR RNA sequence. In green, we actually also show the addition of 2'-fluoroRNA modifications, which is something a lot of our competitors like. We actually particularly don't use it, and we actually don't like it that much because it jeopardizes the activity to a much greater extent than 2'-O-methyl modifications, which is also a modification that is naturally occurring in organisms. So we did the same walk here and tried to come to consensus of sequence to increase the stability of our sequences. And what we learned from this modification walk is that when modifying some, it can lead to severe loss of function, and we learned that some bases are more editable than others. And so based on the entire study here, we actually came to a compromise. We came to an optimized format, which is the one that we now have available as the commercial solution, the Altar CRISPR system. I'm going to talk a little bit about the delivery of the optimized reagents that we have, the Altar CRISPR system. The idea is basically to deliver the three components of the CRISPR in one go, and that means the CRISPR RNA, the tracer RNA, and the protein. So first step is that you make a complex, so the initial complex that you made is between the CRISPR RNA and the tracer RNA. You then add the ribonucleoprotein, and all this is actually in equimolar amounts. So you need a little bit of incubation time to optimize the formation of the both complexes, but then the RNA complex can be delivered directly into the cells. So in a relatively short amount of time, and actually in a very easy way, you have the entire plug ready to be used. There are a lot of ways of actually delivering the complex to the cells, and actually we've tried a variety of them. So first there is the cationic lipid transfection. It is a really cheap and efficient method for all the stable cell lines, like most of the common model cell lines actually. We have tested a lot of these cationic lipid transfection reagents, both commercially available and non-commercially available. At this point we would say that the only two that we found to work reasonably well were lipofectamine RNAi Max and lipofectamine CRISPR Max, both from the company Thermo. They're very efficient actually at delivering the ribonucleoprotein, the RNP. We don't know yet what the exact mechanism is, but it works really well. And another advantage that we saw was that the toxicity is not really an issue, even less with CRISPR than with the RNA IMAX, although both gave similar efficiencies. For more complex cell systems, there is the other option, electroporation. Now we have some experience with the Lonza system called the nucleofactor, which was previously from the company Amaxa, and also some experience with the neon transfection system from Thermo. Both work reasonably well. The difference is that the Lonza system is a black box system with one protocol, limited possibility to optimize the workflow actually. Now, if necessary, we can advise you a little bit on how to optimize this system, so you can get in touch with us afterwards. The Neon system, on the other hand, is an open system with a lot of parameters that can be adjusted, but the drawback here is that you have so many parameters that can be adjusted that you might encounter difficulties to reach the sweet spot of the delivery of your complex. Now, one of the questions that we often get is now, how stable are those RNP complexes? So we are right now in the process of starting a study to measure the stability. We now have data that we show here is data for 2 weeks, stability study 2 weeks at 4 degrees, minus 20 degrees, and minus 80 degrees. And we see that there's hardly any degradation, not to say that there's actually no degradation at all, so it's very stable. We just actually got internally the data of 4 weeks, and that's actually where we start to see a little bit of degradation at minus 20 degrees only compared to the initial values. Our guess is that this difference is actually initiated by the several freeze-thaw cycles that the complex is, that the complex is going through. So at this point, we're pretty confident to say that we can keep them at least 1 month at 4 degrees or minus 80 degrees. But the thing to take into account is to best limit the freeze-thaw cycles as much as possible. So 4 degrees is a good choice, of course, unless you want to store your complexes long term. On this slide here, we are comparing this different sources of Cas9. What we're doing is comparing the delivery of the CRISPR system with our own two-part CRISPR tracer RNA system existing of synthetic sequences, but where the Cas9 is delivered in the first instance as a plasmid, second here as mRNA, or as the ribonucleoprotein complex. And we compared that with what we call the gold standard being a stable version of a Cas9 expressing cell line. Unfortunately for that, we only have one for humans, so not for rat or mouse. But what you see is that the potency and the efficiency using the RNA complex comes very, very close to the best case scenario. So how is that explained that when you use plasmid or mRNA, the efficiency actually drops drastically? This is partially due to transfection efficiency, but it's also due to the efficiency of the process in itself. What I mean by that is that in those two cases, the Cas9 needs to be expressed and needs to migrate to the nucleus to be active. By that time, it can take 48 hours. Some of the RNA might already be degraded or might be sequestered and not available for the formation of the complex anymore. So bottom line, what this slide here shows is that it's better to provide the entire complex of the sequences and the protein to the cell immediately at the start to get the highest efficiency. This here is an observation in a publication, so it's no data that we obtained, it's from this publication here in the Journal of Biotechnology. What they did was really to highlight the advantage of using protein, and that is because the protein actually doesn't stay that long in the cell. Actually, within approximately 72 hours, the protein is pretty much completely gone. The mRNA, on the other hand, stays much longer, and the plasmid can actually stay in the cell for several weeks at very high level. The authors here managed to correlate this to the off-target effects. So what they did here was they looked at those two sites. Now we didn't have the chance to reproduce these results yet, but actually, when you think about it, totally makes sense what you obtain here, because the more Cas9 you have in your cells for a longer time, the higher the chance of having those off-target insertions and deletions. So just to summarize here, we call our approach the commando approach. And what we do is, in the commando approach, is we immediately deliver the entire complex to the cells so that you don't need to wait for one of the components to be expressed over time. It's active from the start, it acts very quickly, and then it disappears. We're going to finish the presentation with a couple of examples from our end users, all of them with permission of the end users. Now, some of them were not comfortable of having their name on the slide, so mostly corporate customers were not comfortable with that. A lot of pharma companies have adopted our two-part system in combination with the protein. This slide here is from one of the top 5 pharma companies. We're actually working with them towards a human genome library, so to give researchers the opportunity to test the entire genome. They work with hematopoietic stem cells, and what they did was to target one specific gene and then look at the increased expression from another gene. So the data shows actually the higher expression in the gene they're monitoring. They compared our ALTAR system delivered with the RNP complex with one of our main competitors, and for all the sites they see a favorable effect in using our approach. Being a top pharma company in the Bay Area, they've been in the CRISPR research from a very early stage and have tested really all the possible technologies. And we got the extremely nice feedback from them that it was way better than anything they had tested before. Just because of the combination, again, of the protein with the optimized and more stable shorted sequences. Also, it's pretty hands-off. You don't need to perform any IVT. You don't have to clone. You just need to order those products and use them as they are. This slide here is an example in zebrafish from the Shear Lab at Harvard University. What they do is micro injections of the RNP complex. And so you have a very clear phenotypic effect with a lot of pigmentation and some other modifications of the embryo development as a result of introducing the RNP complex and targeting a specific gene. You can also see that the number of embryos that were affected and transfected are given over here. And so here is the non-targeting CRISPR reagent and the targeting ones. And so the high incidence of animal show the adapted phenotypes. So this is actually much higher than most publications will show you. This example from the Kramer group shows the utility in C. elegans. Again, this is data from an external source. What they do is they do micro-injections of the C. elegans with Cas9 as RNP. They're looking at the formation of the dumpy left roller phenotype. In addition to testing different ways to generate the modified worm, they tested 2 versions of our CRISPR RNA, the modified and the unmodified ones. And so when we approached them, they also tested the single guide RNA made by IVT. In C. elegans, they were very successful using that, but it came with a lot of work, making the IVTs less favorable. And also, when you have IVTs, you don't know exactly if the reaction works. So if you have that full product, there is a lot of bias that can be entered in your experiment. And so bottom line, overall, the workflow, from a workflow perspective, they found it beneficial to move over to our system. And as you can see, the modified sequences of our CRISPR RNA gave much higher efficiency compared to the natural occurring unmodified CRISPR RNA. So to conclude this presentation, we just want to quickly highlight what we offer. So we have since recently the Cas9 endonuclease available. One of the major issues for people to use Cas9 was actually, until we launched it to the market, the price was extremely high. So what we did was to come into the market and make it much more affordable. So our competitors have actually been quite greedy on this. We price it at levels which are up to 15 times more favorable from a cost perspective than what was currently available in the market. We realize that when you market at the price that we do, more people are eager to actually adopt it, because at a high price only a selected amount of customers will use it. But we are strongly convinced that the benefits of using the RNP system with the optimized sequences, and so we would prefer to have that available to all the end user to be able to perform genome editing effect with higher efficiency than with other systems available currently. So we also, of course, have the chemically synthesized CRISPR and tracer RNA available. We're currently in the process of developing a design tool that will offer on the website probably in the next 3 to 4 months. We have the universal tracer RNA. We sell them in separate tubes so that you can actually order a larger amount of tracer RNA depending on the amount of reactions that you want to do. We have controls for human mouse available as well. That will allow you to check that in your system things are working before you really spend money on designing design complexes. So it's a great help, and just for your information, if you need help from a technical point of view, feel free to reach out to us. So we have a lot of information available on our website when you visit www.idt.com/CRISPR. There are protocols, there's technical questions that can get entered. And so I would like to thank the scientists who contributed to these studies. So they're all part of our CSO group, Dr. Mark Belke, Ashley Jacoby, Gareth Redding, Michael Collingwood, Michael Christodoulou, Molly Tiernan, Sarah Ziner, and Kylie McQuiston.